know that the dream of every cancer patient is that when they have this tumor excised, they could take the tumor cells from their tumor, grow them in some type of dish, and we could screen cancer therapeutics and actually know which kind of chemo is going to work for them, because chemo is so exhausting. It's so hard on the patient. Now, we're a long way from that, but in the shorter term, what we want to do is be able to identify or create a platform that is able to identify better candidate drugs. Because so many candidate drugs are identified at the 2D stage that don't work in the animal stage, or at the animal stage that don't work in a human. And so if we have a way that we can use patient-derived cells or patient-derived xenografts that could, better, screen out the things that aren't going to work, so that we can get to the things that are going to work. So in our lab, we're interested in creating biomimetic models outside of the body that reflect what's going on inside the body. And one of the areas we're working on is engineered tumor models. And so in that case, typically in screening cancer therapeutics, there's 2D cell sheets, there's cell culture models, and then there are animal models and eventually clinical testing. But the problem is that 2D cell sheets really don't reflect what's going on in the animal models, and the animal models don't really reflect what's going on in the human body. So our group is working to make basically an intermediate step, creating 3D engineered tumor models in a dish that are going to better reflect what's going on in the human body. We've worked with breast cancer cells, we've worked with colon cancer cells, we've worked with prostate cancer cells, for a range of research projects across campus in collaboration with people in the School of Pharmacy, in the School of Nutrition, and in the vet school, to look at how manipulating the tumor microenvironment, it manipulates a tumor cell phenotype, and how that may or may not reflect what is happening in the animal model and in the human system. By creating engineered tumor models in our lab and looking at different cell types and different types of cancer, our end goal is to create a model that reflects what's going on in the body to the greatest extent possible. And when we can do that, then eventually what, as a field, we'll be able to do is take patients' cells from their tumor, put them into the model, use that for drug testing, and more effectively identify which chemotherapeutics are going to be useful for that patient, particularly in metastatic disease.